Phototransduction is the conversion of light energy into electrical activity that ultimately terminates at the primary visual cortex of the occipital lobe of the brain. And it's the responsibility of a number of different cells, photoreceptor cells, some excitable cells, and neuron type cells. So in this slide, what we have right here in green is a photoreceptor cell. What I've drawn here is certainly a rod. Rods and cones are the photoreceptor cells, but for our purposes in this demonstration, I'm not really concerned if this is a rod or a cone. This is going to be a very simplified version of what happens within the retina of the eye. The retina is the innermost layer of the eye that contains these photoreceptor cells. So once again, photoreceptor cell in green. In the middle, we have a bipolar cell, and at the bottom, we have what's known as a ganglion cell. And the axon of the ganglion cell will run in the optic nerve, which once again terminates in the primary visual cortex of the occipital lobe. So let's take a look at this. In the dark, the photoreceptor cell has a voltage of negative 40 millivolts. Now, every book, every professor I've heard speak about this, experts that speak about this, describe this cell in the dark as being depolarized. And I still can't accept that unless the exterior of the cell is negative, which it's not. As a result, I would suggest that this cell is excited, but it's certainly not depolarized. It's at negative 40 millivolts. And the reason we're at negative 40 millivolts instead of our typical negative 70 is because we have cation influx. We have sodium and calcium moving into this cell. At the same time, we have potassium leaving, but because we have more sodium and calcium moving into the cell becomes excited or less negative. Other things to look at in this cell, in purple right here, we have what's known as a light sensitive protein or light receptor protein that gets activated, undergoes a conformational change when it's activated or energized by photons or light rays. Connected to that is this red structure known as transducin. Transducin is a G protein, which means this whole setup that I'm going to describe here involves a G protein linked receptor. This light receptor cell right here, this protein, is the receptor cell, and transducin is the G protein. So I've mentioned G protein link receptors in previous lectures, but I haven't talked about them in detail. What I have suggested, though, is that G protein link receptors, when activated by a ligand, by voltage, by some sort of mechanical manipulation, or in this case, light, it initiates a whole cascade of events. So muscarinic receptors for cholinergic receptors, muscarinic receptors, when bound by acetylcholine, initiates a cascade of events because muscarinic receptors are G protein link receptors. The adrenergic receptors, alpha and beta, are both G protein link receptors. So this is a nice prelude into how G protein link receptors work. It's not an extensive overview. It's fairly simplified, but it will suffice for our purposes. In orange, we have an enzyme that transducin will eventually activate. It's not active in this picture right here, but that enzyme is known as phosphodiesterase. One last thing we have here is cyclic GMP, which we see bound to this channel. When there are high levels of cyclic GMP, it keeps that channel open, allows the influx of those cations to excite the cell to bring it up to negative 40 millivolts. When that cell is excited, it releases its signaling molecule known as glutamate. When we've talked about glutamate, we've suggested that it is an excitatory neurotransmitter in the central nervous system. Keep in mind, we're talking about the eye, And while it's very close in proximity to the brain, to the central nervous system, it is not part of the central nervous system. So glutamate in this situation is a signaling molecule, but it's not specific to the central nervous system. And the reason I highlight that is because in this scenario, this glutamate is going to function as an inhibitory signaling molecule, not its normal excitatory. So when that glutamate is released, it binds to the bipolar cell and inhibits the bipolar cell. That is to say, there's no activity in the bipolar cell. If the bipolar cell is inhibited, it does not release its signaling molecule, and the downstream ganglion cell is inactive. Nothing's going on with the ganglion cell. So everything we just described was in the dark. When that light-sensitive protein gets activated by photons or light rays, it liberates this G protein. It used to be linked to this receptor. When that G protein is liberated, it activates phosphodiesterase, which cleaves or hydrolyzes cyclic GMP, which means we have low levels of cyclic GMP in this cell. Cyclic GMP is no longer bound to this channel. If cyclic GMP is not bound to the channel, we no longer have sodium and calcium influx into the channel, but we still have potassium efflux. So now the cell has hyperpolarized down to negative 70 millivolts. And when it's in this state, it does not release its inhibitory signaling molecule known as glutamate. Because it doesn't release its inhibitory signaling molecule, the bipolar cell becomes excited, it depolarizes, and releases its signaling molecule. And just to confuse you, this signaling molecule is glutamate, but it's going to act as an excitatory signaling molecule this time. And really the big difference is not in the signaling molecule, it's in the receptors. And I'm not going to get into that in this video, but receptors are the ones that are really dictating what impact signaling molecules have. So once again, this bipolar cell is activated, it is depolarized, it is excited because it's no longer being inhibited superiorly by glutamate. This glutamate released from the bipolar cell will activate the ganglion cell. This ganglion cell will depolarize and send a signal down the optic nerve to the primary visual cortex of the occipital lobe and allow us visual sensation.